Hello, everyone. Thank you for attending today's webinar, Guidelines for Developmental and Reproductive Toxicity Testing, presented by Frontage Laboratories. I'm Andrea Anderson, and I'll be moderating this webinar. Our speakers today are Bjorn Thorsrud, PhD, Vice President of DART and Scientific Operations, Frontage Laboratories, and Supita Monacal, PhD, DABT, Director of Toxicology and Technical Operations, Frontage Laboratories. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. All right, let's begin. Supita, please go ahead.
Hi, thank you, Andrea. Thank you, everybody, for joining us. The topic of the webinar, as Andrea previously said, is developmental and reproductive toxicity testing. So we're going to talk a little bit about the regulatory guidelines that are required for these DART studies, and the overarching goal is to describe the FDA requirements of the initiation of preclinical DART studies using mammalian research models. And in order to initiate clinical trials for women of childbearing potential, these studies need to be performed. These studies provide very important information about the effects of drug exposures prior to and during parental mating, as well as looking at maternal and fetal changes that may occur during gestation and looking at any alterations in the development of the offspring following birth. So as a quick outline of what we'll go over today, we're going to give a little bit of a historical perspective on DART studies and how they came about. I'm going to talk a little bit about critical windows of susceptibility, as well as risk assessment. And then Bjorn is going to hop on and talk about DART study designs. He'll review 3 typical designs, and he'll give a summary of those designs and some timelines for dosing and completion of the studies. And then he'll talk a little bit more about other aspects of DART study designs and other types of designs, as well as the direct guidelines and SEN conversion requirements, as I'm sure we all know and love. So to begin, I thought I would give perspective on where DART regulation came about. So I'm sure we all know of thalidomide, which was a drug that was developed in the 1950s to treat morning sickness. It was thought to be very safe, non-toxic, and all of the initial preclinical testing was done in rats. However, after some widespread use in Europe, Japan, Australia, thalidomide was found to be a potent teratogen, and it affected over 10,000 babies in Europe, Japan, and Australia. Some of the major findings included phocomelia, which is a congenital deformity where the hands and the feet are bound to the child's trunk, or they're absent or grossly underdeveloped. And there's an image here showing that. Ultimately, when these congenital defects were identified, and unfortunately after children were already developing these malformations or being born with these malformations, thalidomide was taken off the market in 1961, and more testing was done, and they found that in preclinical testing in rabbits, the teratogenic potential of thalidomide was confirmed. And as a result, now this spurred the FDA to assemble a committee of scientists to develop guidelines to assess the hazard potential of new therapeutic agents for developmental toxicity, especially if the intent is to go into women of childbearing potential. And another note was that toxicity testing needed to be done in one other species aside from rodents, and rabbits were chosen. Now, the thalidomide incident and everything that came out of it led to a subsequent expansion of teratology and developmental toxicity. In this period, fetal alcohol syndrome, which had been around for a while and relatively well characterized. It was first recognized and described during this period following the thalidomide incident. So this is 1968 to 1973, and as I'm sure most of you know, fetal alcohol syndrome does have very characteristic craniofacial malformations. We've got a picture here showing that in a mammalian species as well as in a human. Another example of a teratogen is Accutane, which is 13-cis-retinoic acid. So it is commonly referred to as Accutane embryopathy. When a mother is using Accutane during pregnancy, the fetus will have a characteristic pattern, or the baby, when it's born, will have a characteristic pattern of mental and physical birth defects due to the use of this retinoid, which again is a synthetic vitamin A derivative. The most characteristic defects include hydrocephalus, microcephaly, and then intellectual disabilities, ear and eye abnormalities, cleft palate, other facial malformations. And just a couple more examples. Another pharmaceutical, diethylstilbestrol, or DES, was prescribed from 1940s to the 1970s to prevent pregnancy loss. And here's an ad from that period of time. It was very widely propagated and used. And about 25 years after its introduction into women's healthcare, it was confirmed to be a transplacental carcinogen and teratogen in humans, which is very unfortunate. And it turns out that DES also has widespread transgenerational effects. So granddaughters of women who use DES show malformations or abnormalities in reproductive function. And then I wanted to also provide one other example that isn't a pharmaceutical, and that's methylmercury. So it's mercury that then was processed to methylmercury. It's an environmental contaminant. Probably one of the most characterized events of methylmercury poisoning was in Japan, and it was called Minamata's disease. This happened in the 1950s, and this was acute poisoning. And adults that were exposed to high levels of methylmercury showed sensory disturbances. It's called glove and stocking syndrome. They had ataxia, constriction of the visual field, auditory disturbances, and tremors. And then children of affected individuals were born and had low birth weight, microcephaly, cerebral palsy, or cerebral palsy-like symptoms. And not only that, even at low doses of exposure, there are subtle neurobehavioral developmental deficits as well. So even if there aren't acute outright symptoms of poisoning, you might have children that are born with other behavioral or neurological deficits. So that being said, as the field continued to grow, there was an assessment of critical periods of susceptibility. Now, the idea of these critical periods or critical windows has been around for a while, since probably the late 1800s. And the idea is that there are windows in which different organs and different aspects of development may be affected depending on when exposure occurs. And so these two images here kind of show the developmental system and where different organs and different tissues might be differentiating, growing, and where altered development could be affected. I'll give you a second also to look at this. And when Bjorn talks a little bit more about DART studies and the dosing paradigms, you'll see that the timing of exposure is critical in these tests. So to get a little bit more mechanistic, one of the biggest challenges of risk assessment is identifying a mechanism of toxicity for these drugs or these toxicants. Normal development is extremely complex. So when you're working with an adult that is fully developed, that mechanism of toxicity is more or less known, but not always clear. But when you're talking about exposure during periods of development where there are widespread changes occurring, that adds another level of complexity. So a toxicant can interact at numerous different critical pathways and lend to developmental toxicity in a number of different ways. So identifying how a drug might be affecting fetal development is pretty difficult and pretty challenging. Another point is that toxicants, especially environmental ones, may not be one single pharmaceutical ingredient. It could be a wide range of chemical, physical, and biological agents that can initiate a wide variety of mechanisms. So you might not just be affecting one receptor or one level of organ development. You can be affecting several different parameters. And then, of course, some toxicants might affect only a fraction of individuals in a population. So this could be related to genetic differences and, of course, differences in health history. So diseases, nutrition, other exposures, and other propensities. So the potential action of a toxicant has to be understood at a lot of different levels of biological organization. So not just at the cellular level, but also across different pathways. And in this way, we can start to understand the overall process of developmental toxicity. So that leads into risk assessment. Of course, within the realm of toxicity testing, DART studies, developmental and repro tox studies are not separate from the application of general toxicologic principles. So a lot of those principles still apply. We're still interested in toxicokinetics, pharmacodynamics, ADME, all of those parameters. But the difference is there needs to be specificity or special emphasis on the timing of exposure and the duration of exposure. So going back to those critical windows of susceptibility, because development in all different aspects continue to occur during fetal development. The dose level, what may be a dose level that is chosen in a mature adult may not be the same dose level that you would want to choose for a developmental and repro tox study. So that is, and Bjorn will go into some of the nuances of choosing dose levels when it comes to performing a DART study. And I think most importantly, you need to consider the consequence to the unit. So you're not just looking at maternal toxicity, but you're looking at the maternal and fetal unit. And with that, I've given a bit of that historical background and some examples of potent teratogens in our environment and also in the pharmaceutical industry that have been assessed in the past. We've talked a little bit about critical windows of susceptibility and some challenges in risk assessment as it relates to mechanism of toxicity and dose level choice. So with that, I'm going to turn it over to Bjorn, who will dive deeper into actual DART studies. Bjorn?
Thank you so much, Peter. You did a great job providing us a good perspective and, excuse me, the importance of timing when drugs are being exposed to embryos during pregnancy. I'd like to maybe give away my age a little bit, but the period of the thalidomide tragedy happened to be exactly the years that I was actually born in Europe. So it's kind of interesting that I'm actually become a teratologist looking at this field, going back through that. So it's really, really important that we take care in making sure that we don't just use adult dosages to women of childbearing potential, because there are a lot of things that are going on, not just in the maternal system, but also in the embryonal, fetal, and pup processes. Right now, we have the ICH guideline. It's the International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use. It's a long name. And there's a specific guideline that's called the S5R2, detection of toxicity to reproduction for medicinal products and toxicity to male fertility. And this essentially are regulations that are relevant to the United States, Europe, and Japan. The ICH S5R2 guideline was issued in 1992 and updated in 2005. And the most recent updated guideline is now effective July of 2020. So we are right now under the ICH S5R3 guideline. I will be providing links to these guidelines if there's interest after the talk. I'm going to mainly focus on in vivo designs, rat and rabbit designs for DART studies, which are the most common species for this type of testing. There are other research models available as well for DART testing, which can include the mouse, hamster, dog, mini pig, and monkey. Okay, let's get to the study designs. There are essentially three that I'd like to cover during this presentation. One is the embryo-fetal development study in rats and rabbits. This is also known as a SEG2 study or a developmental toxicity study. There are many different names that I'm using the guidelines abbreviations to discuss these studies. The second one will be the fertility and early embryonic development study in rats, which is called a FEED study, which is kind of a strange name to it because it's not really a FEED specific study. And the third type is the pre and postnatal development study in rats, which actually looks at what happens to the offspring after they've been born. Okay, let's start with the embryo-fetal development study. Again, the aim of these studies is to detect adverse effects during pregnancy in the female and to look at the development of the embryo and the fetus during this period of time or during organogenesis period. And there are specific sensitive periods, as Sapita pointed out, that we need to keep in mind when going through that. So this is key in doing these studies is getting the timing at the right time to make sure that we can assess potential risks during pregnancy. For this type of a study, rats are usually dosed between implantation and closure of the hard palate between gestation day 6 and 17 in rats, and in rabbits, which is the second species, which is really important to have since Sapita pointed out for the thalidomide, had they had tested it in rabbits, they would have found thalidomide to be a teratogen. Then we have C-sections on gestation day 21 in rats and in rabbits on gestation day 29. Here's a little picture to maybe make it a little bit easier to look at. We receive time-needed animals. We start dosing them on gestation day 6 at implantation and continue dosing them all the way to closure of the hard palate, which is gestation day 17 in rats and gestation day 18 in rabbits. Then we stop dosing and we evaluate the animals, the pregnant animals, on gestation day 21 in the rat and on gestation day 21 in the rabbit. For dose range finding studies, we only do an external fetal evaluation in comparison to the definitive studies where we do external visceral and skeletal evaluation. It's a lot more thorough. Let's talk a little bit about, I said, dose range finding studies. These are very important to figure out what the drug may be doing to a pregnant animal. It's usually a smaller study containing about 5 dose groups, 1 control and 3 treated. We test about 5 to 8 pregnant females per group, and the high dose should show some toxicity without lethality, and the low dose should show no adverse toxicity. Again, we look at standard maternal clinical observations, body weight, and food consumption, and then we'll do the C-section on gestation day 21 in the rats and on day 29 in the rabbits. We look at the dams or the does, depending on what species you're looking at, to see how the drug affected the pregnant female. And then we also look at the fetuses at C-section to see if we see any external changes in these animals, as well as survival of the animals at that point. We look at implantation sites. We look at pre and post implantation changes, which means that we want to find out if there were any deaths during the dosing period for these animals. Next, we use this information from these dose range finding studies to select doses for a definitive study. And a typical definitive study will have 4 dose groups, 1 control plus 3 treated. There are usually 23 time-mated females per group. Again, the high dose should show some toxicity without lethality. FDA will not accept a study that doesn't show some toxicity. So we always want to make sure that we do have some toxicity, such as, for example, a decreased food consumption, body weight, 10% decreases in those parameters, for example. And the low dose should have no adverse toxicity. Again, we look at the female for standard clinical observations, body weight, food consumption, and we do the C-sections, at which time we find out what was going on with the fetuses, where we do both external, visceral, and skeletal evaluation. This is a very thorough examination to make sure that there were no, if any changes in any of these parameters can happen. As Peter mentioned, there are susceptible periods, but there are also susceptible tissues during the gestation period. So we can then find out to see if any of these processes were affected by the drug in question. The next study is a fertility and early embryonic development study. And the aim of this study is to test for effects resulting from treatment that's initiated prior to mating of males and females, and then continued through mating and implantation. And in this case, we don't run dose range finding study. We go right into definitive studies. Males would be dosed for 4 weeks prior to mating, through mating, and until confirmation of pregnancy by the females. And the females are dosed 2 weeks prior to mating, through mating, and up to gestation day 7, which would be implantation. Again, we have 4 groups with 25 animals per sex per group, with 1 control and 3 treated animals. Again, the same motto applies. High dose should show some toxicity without lethality, and the low dose should show no adverse toxicity. In the males, we also look at the sperm evaluation. We look at count, morphology, and motility to see if we see any effects of the drug on the males. For the females, we look at the implants to see if we see any changes. Were they implanted? Were they resorbed? To look at the pregnancy outcome and potential fertility. Does this drug affect the ability for a conceptus to be fertilized and be implanted? Here's a quick figure showing how we do this. Usually we get animals in. These would be non-pregnant, prima gravida animals that are dosed, again, for males 4 weeks prior to mating and females 2 weeks prior to mating, all the way to gestation day 7 to implantation. These animals are then mated, and most of the time the males that are treated are mated to the treated females, and then we look at the effects at C-section on gestation day 13, and we look at the males at confirmation of pregnancy. So we get an idea of what's going on. Now, we could potentially have another design where we would be dosing sexes individually, or a cross-linked design where you would treat males and mate them to untreated females, and vice versa to the females, to see if there are any male or female mediated events taking place. The last one is the peri and postnatal development study, and the aim of this study is really to detect effects following exposure during following implantation all the way through weaning. So this is a long period of time. The offspring is not being dosed. It is the dams, the pregnant female rats, that are being dosed during this time. Again, we usually only run a definitive study. No dose range finders are required, where we again dose the animals from implantation to the end of lactation. And as I mentioned, exposure to the embryos or fetuses or pups would be in utero during gestation or ex utero during lactation. Again, figure to kind of give you a perspective on how this is put together. We have get the animals in, we can mate them in-house, or we can actually get time-mated animals that are received, and then they start dosing on gestation day 6 at implantation. We go through, the pups are delivered, and the dams are continually dosed until the end of weaning on lactation day 21. During this time, we look at these pups for different scenarios, and I will describe that next. We look at animals during the lactation period when they're still nursing with the dam, and we look at to find out what types of changes we can detect in these animals. Then after weaning, we select F1 animals, the offspring, to be, we usually have one animal per sex per litter that's selected to continue on study. And they are then looked at for sexual maturation to see if there's any effects on the animal's ability to reach sexual maturity, looking at preputial separation, the vaginal opening. And we also do behavioral testing, such as Morris water maze for learning and memory and motor activity. These animals are then mated when sexually mature, at least 10 weeks old, and we then take the F2 generation down on gestation at 13 to confirm pregnancy. So we've actually gone from the parental dosing of the females from gestation day 6 at implantation all the way to the end of lactation. Then we take those animals, are selected to then continue and are being tested and also mated to see if they can actually produce a viable progeny. Okay, so these embryo-fetal development studies and the feed studies can run in parallel and are generally required for phase 2 clinical trials. Things are changing with some of the guidelines to make sure that women can be properly represented, but usually we like to have preclinical testing done before women of childbearing age would be exposed to any new chemical that's not been used before. For the PPND study, it's usually required for phase 3 clinical trials, but they can also be done earlier to make sure. So it all depends on the compound and the population that would be affected at this point. So again, these designs provide a comprehensive evaluation, as I discussed, for drugs, for in vivo testing to see if the animals can conceive a viable conceptus. Essentially, can they be fertile? They can be then evaluated during pregnancy and lactation to see if these effects are showing any type of teratogenic or fetotoxic effects. And then we can actually look at the postnatal development of these F1 animals and see if they can truly produce another F2 generation. Here's just a quick chart that may be helpful to you. This shows all the different types of studies, the guideline, routes, species. The routes can change. It doesn't always have to be oral, but this kind of give you a general idea of how many animals are used, how many groups are used. TK animals are usually only done in embryo-fetal development studies, and it also gives you a little idea of the approximate in-life length and report phase typically for these studies, and if they're GLP compliant or not. Some additional information. It's important to note for embryo-fetal development studies, especially in oral formulations in rabbits. Rabbits have a tendency to be quite sensitive to changes in their GI system. And aqueous solutions usually work quite well, in contrast to lipophilic types of solutions that can affect the GI tract. The animals can sometimes stop eating and then abort, and now you don't know if the effects that you're seeing are due to the animals not eating or if the animals are aborting because of direct effect of the drug. So sometimes you may have to do a different route of exposure in rabbits to avoid the GI tract. We always recommend using TK or blood collection to look at systemic exposure in the animals and dose range finding studies too, so that we can properly select dose levels for definitive studies. And one more thing, since rabbit studies are usually not very commonly done during the standard preclinical general tox profiles, it's sometimes not a bad idea to just run a study of non-pregnant animals, kind of a tolerability study, just to make sure that the animals can tolerate the drugs at the recommended dose levels. As I mentioned before, for the fertility studies, the feed study, you can do a combined study, which is standard, or we can do a crossover design where the treated males are mated to untreated females, and then the animals, the treated males, will be mated with the untreated. The treated females will be treated to the untreated males. Sorry about that. Generally, TK is not required for fertility studies, as most of that type of information has already been obtained in previous 28-day general toxicity studies. For the peri-postnatal studies, we usually cull pups on lactation day 4, and the reason we do that is to make sure that all the females are nursing the same number of animals, so you don't have one litter that has 12 and one litter that may only have 2, because that could definitely affect the size of the pup and the effects potentially to the effects based on nursing practice during this time. Milk collection for test arc evaluation is possible. It's not commonly done, but if it is required, we would recommend satellite TK animals be used, and we would collect blood samples from both the mother and the pups mid-lactation. Since the amount of blood may be fairly large, you may have to pool those samples on a litter basis. But this provides you very valuable information. Make sure that whatever drug the mother is taking in has also been passed on to the pups. Because if we only do a milk collection, we could determine that yes, there's drug in the milk, but we would not know how much it would be in the pup. So this way, you can actually compare the two and see if there are any changes and if the pups are being affected by the drug. There may be combination studies that could be done. Trying to save time and money sometimes, you can combine some of the fertility and embryo-fetal development studies, or you can combine an embryo-fetal study with a peri-postnatal study. It all depends. These things can be done to try to save some time, but ultimately the designs I described are fairly robust and really provide good information in terms of what is happening so that we know specifically which time period the effects took place, instead of making a combined study where you have a much longer dosing period and you may not know exactly when the effects actually took place. A fertility component may also be added sometimes to standard general toxicity studies to see if there were some effects previously seen in the reproductive organs. That is possible. There's a PEFT study that's a new design in the updated guideline. It's similar to a dose range finding embryo-fetal development study, but has a small number of animals, but it also requires visceral evaluations, as is required in the definitive study. And just want to let you know that PEFT studies are used to, if it's used to defer a definitive embryo-fetal study, then this study should actually be done in accordance to GLPs. TK data should be collected from the dams, and skeletal evaluations should also be performed. So to me, I like the idea of doing dose range finding studies and instead of just jumping right into a PEFT study. However, if a clinical program is already ongoing, that may be an option to use to be able to defer the definitive study. Here are some links to the ICH guideline. One's the FDA guideline. They use the same guideline, but they have a slightly different format. The other one is the European Medical Agency, EMA. Now for a question a lot of people have, and that's SEND conversion. I know that everybody is worrying about, okay, do I have to do SEND conversion for my DART studies? Right now, it is not required until the 15th of March of our studies starting before after the 15th of March 2023. However, there are a lot of companies that are actually running SEND data sets at this point, just to make sure that everything's all set, and they are subcontracting this type of work is actually being done right now, because the portal is open at the FDA to be able to run it. But again, these DART studies are not required until the 15th of March 2023. There's also a really good book that I would recommend that I always reference, and that's a book by Ron Hood, Developmental and Reproductive Toxicology, A Practical Approach. There's now a third edition available, and it provides some very valuable information on both the basic designs of developmental and reproductive toxicology all the way to the details of all the different types of findings that can be looked at during a fetal evaluation. So I would highly recommend this book if you have additional questions on developmental and reproductive toxicology. That's it.
Thank you so much for the presentations. We will move on to the Q&A now. There is still time to submit your questions using the Q&A tab to the left of your screen. We have a lot of great questions already, and we will try to get to as many as possible. For the first question, do all CROs perform DART studies?
Yeah, let me take that question. No, all CROs do not perform DART studies, and you should probably take great care in looking at a facility that has a long history of running these studies, because these studies require specialty care with specialized technicians as well as study directors that are used to looking at these specialized studies. So I would definitely recommend that you look at CROs that do have history running DART studies.
All right, another question. Do you need a historical control database?
I'll take that one too. Absolutely. I think that's another part of the importance of looking at a lab that can do DART studies. They need to have a historical control so that you can distinguish normal development where there is some abnormalities that just happen in control animals, and that is really, really important, so you can distinguish the possibility of a finding that may have just normally occurred instead of a problem with due to the test article itself. So historical control is another key component, and there are some online databases that are available as well too.
Okay, next question. Are combined study designs possible?
Just as I mentioned, I briefly mentioned this, you can do combined studies. Sometimes that can help in timing, trying to get things done a little bit quicker. But ultimately, the three designs that I discussed have some advantages by really providing a very robust view of exactly what is going on at the specific time points that you're looking at. Because when you're combining studies, you may be dosing over much longer periods of time than you would for a single study. So I would probably recommend going the standard design, looking at each one of the three over time, but it is possible to do combined studies, and sometimes the FDA may recommend that as well too. And obviously, then you would proceed with such a design. Thank you.
We have time for one more question. We had a lot of great questions today and couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. So for this last question, are rats and rabbits required for all DART studies?
Okay, I'll take that one as well. No, rats and rabbits are only required for the embryo-fetal development studies. The feed studies, the fertility study, and the pre-postnatal studies do not require rabbits. So to answer the question, yes, you do need to have rats and rabbits for the embryo-fetal development studies. Thank you.
Thank you for those answers, and thank you all for attending this Fierce Biotech webinar and for submitting so many great questions. I'd like to thank our speakers for participating and Frontage Laboratories for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.